discuss today about molecular diagnosis. In the previous lecture, we discussed human insulin production. In the other lecture, we discussed gene therapy. Now, our today's topic is molecular diagnosis. 12.2.3, right? It is 12.2.3. It comes under molecular diagnosis. I think we know what is meant by diagnosis. Diagnosis means detection, right? So, what is molecular diagnosis? How is this different from conventional diagnosis? Let us try to understand. Now, diagnosis, when we go for a diagnosis means if there is an ailment, if there are some symptoms, if the body is showing some symptoms. So, to confirm whether the illness is there or it's not, so we have to go for a diagnosis, right? So, then if a person is diagnosed with a positive report, then the doctor has to start the treatment immediately, right? So, effective treatment of any disease depends on early diagnosis. If a patient is diagnosed early, then the concentration of the antigen will be less in the body, then the doctor can treat it effectively, right? The doctor can kill the pathogen effectively. So, we understood that early for effective treatment, So, we are telling that early diagnosis is required. If we want to treat it very early, then early diagnosis is required. But when we go with conventional techniques, what do you mean by conventional techniques? By conventional diagnostic techniques. What do you mean by conventional diagnostic techniques means, where is the pathogen seen? The pathogen is seen in the blood or in the body fluids like urine and all those things. When we go with conventional diagnostic techniques means, either we go with blood estimation or we go with urine estimation. So, we call it a serum analysis or urine analysis. By conventional diagnostic techniques means it comes under serum analysis, or we can go with urine analysis. So, if a person's urine is collected and in the urine, if you are checking the concentration of a pathogen or any particle, right, any biochemical which is present in our body, calcium level should be so and so, iron level should be so and so, cholesterol level should be so and so. So, we need to test that also. Decreased concentration leads to deficiency, excess concentration might be harmful also. So, then for that, what do? We take the urine sample or we take the serum analysis. What do you mean by serum analysis? Means like when we take a blood, when we allow the blood to settle outside, then the blood will clot. When the blood clots, all the cellular part gets collected as a puffy layer, and the liquid content which gets separated out after blood clotting is called as serum. So, when we take, when we collect the blood in a test tube and when you allow it to settle, the upper portion, the puffy portion where the blood cells get clotted up. So, this is puffy portion. What is there in the puffy portion? All the cellular components. All the cellular components will be there in the puffy portion, whereas the bottom portion contains straw colored liquid which is left out after clotting is called as serum. So, the bottom portion will be straw colored, light yellow colored, pale yellow colored liquid will be there, which is called as serum. So, the liquid portion of the blood after clotting is called serum. The liquid portion of the blood before clotting is called plasma. Now, in this serum, pathogens or all the elements will be there. Now, when we take the serum and when we do any analysis, that is called serum analysis, or we can call it a serological test. Okay, so we can call them as serological tests. So, if you want to test the concentration of either a pathogen or any biomolecule in the body, so we have to go with serum analysis or urine analysis. If you are going with serum analysis and urine analysis, that comes under conventional diagnostic techniques. So, these were the techniques which were earlier used. But when you go with conventional analysis techniques like serum analysis and urine analysis, early detection is not possible. Early detection is not possible. So let us try to understand this sentence in a better manner. For example, if I go to a patient, if I go to a doctor and if I am complaining all the symptoms of typhoid, I went to a doctor and I am complaining all the symptoms of typhoid, then what the doctor will do, tell me? First question the doctor will ask, from how many days you are feeling that you are having fever? From how many days there is temperature, they will ask you. Then obviously we will be telling from yesterday or from day before yesterday, I am taking the tablet. Then what the doctor will do, so doctor will give a tablet again, he will tell take it thrice or twice depending upon the temperature. They'll ask us to take it twice or thrice and they'll ask us to meet on the third day. If the temperature subsides, then it's fine. If the temperature is not subsiding, they'll ask us to come on the third day or the second day. Doctor knows that it is typhoid. Doctor knows that it's typhoid because what and all symptoms we are telling are typhoid symptoms only. But actually the doctor is waiting so that the pathogen in the person's body will rise to certain level. If the pathogen concentration in the body rises to certain level in the body or in the blood, then this conventional techniques can be done. Then we can take and we can do viral test. In the viral test, we will get the report. So that means by the time that concentration builds up in your body, by the time you do serum analysis and urine analysis, I think the concentration of the pathogen in our body becomes more. Then that's not early diagnosis, right? So, but then conventional, if we want to do conventional diagnostic techniques, the concentration of the pathogen should build this much in our body. So that's not good nowadays. So that's why we introduce new techniques for better identification of a pathogen, even if it is very less. That is called as molecular diagnosis. So now we are very clear what is conventional diagnosis and what is molecular diagnosis. Molecular diagnosis helps to identify the pathogen in very minute concentrations also. Correct? Did you understand now? So molecular diagnosis helps to identify the pathogen. It helps to identify the pathogen in less concentrations also. In less concentrations, if you are identifying, means it is early diagnosis, right? It is early diagnosis. Now, early diagnosis, if we do, effective treatment can be done and the patient will come out from the ailment or the illness very fast without much damage to the body, right? So, first of all, when do we approach a hospital? When the pathogen starts showing its symptoms, right? Presence of pathogen is suspected when symptoms are appearing. So, we are telling that presence of pathogen. When do we know that there is pathogen? Presence of pathogen is suspected when it shows symptoms. Now, when the symptoms appear, then we approach the doctor. When the symptoms appear, when we approach the doctor. But by the time the concentration of pathogen in the body goes high, after getting the symptoms, if we go to the hospital, NCERT says by that time the concentration of pathogen in body goes high. After the symptoms comes, if we go and approach the doctor, when they do the serological test, by the time the concentration of the pathogen will go high. So that is why when we go with molecular diagnosis will help to identify the pathogen in low concentrations. Then what are the techniques which are available in molecular diagnosis which will help us in early detection? There are three techniques basically. So, molecular diagnosis, first one is called as rDNA technology. Will help us to identify the pathogen in very low concentrations. Next one is PCR, polymerase chain reaction. And the third one is Elisa. The fourth one is Autoradiography. Children, these four comes under molecular diagnostic techniques which helps to identify the pathogen in very low concentrations. What are they? One is rDNA technology, recombinant DNA technology. The second one is PCR. So what does PCR stands for? PCR stands for polymerase chain reaction. We will see now what is PCR. And next one is ELISA. ELISA stands for enzyme linked immunosorbent assay. So ELISA stands for E for enzyme, L for linked, I for immuno, S for sorbent, A for assay. ELISA stands for enzyme linked immunosorbent assay, which is another technique coming under molecular diagnosis. And the last one is autoradiography, where we use radiolabel probes to identify the pathogen. So in this board, what we discussed is, so what is the significance of molecular diagnosis, how it is advantageous over conventional diagnostic techniques. What are the two conventional diagnostic techniques? They are called as serological test and urine analysis, serum analysis and urine analysis. What is serum? It is the fraction of the liquid which is collecting after blood clotting is called as serum. Those tests are called serological tests. But the drawback with the serological test is it can identify the concentration of the pathogen only if it reaches this much level. If it is less than that, it will give negative report, right? So, but molecular diagnosis will help us to identify the pathogen in very minute concentrations. Take a screenshot, then we will discuss with the techniques one by one. Right. So, we will talk about PCR first. Then we will go to ELISA and autoradiography. Now, the same technique PCR comes in biotechnology principles and processes chapter also. So, for detailed content of PCR, so you can kindly check that biotechnology principles and processes video where we get a complete details of PCR. Now here only little bit information we will see which should be taken under molecular diagnosis. The first technique we are talking is PCR. I told PCR is polymerase chain reaction. It comes under polymerase chain reaction. Now we know that PCR is a polymerase chain reaction developed by a scientist called Kary Mullis. Who developed this polymerase chain reaction? It is developed by a scientist called Kary Mullis. Developed this technique. And what it will do, it will polymerize the sample. Polymerization means multiplying, amplifying the sample. It is multiplying the sample inside the body na, outside the body na. It comes under in vitro gene amplification. In the body is in vivo. Outside in the lab, in the test tube is called as in vitro. So PCR is a technique where we will try to increase the concentration of the pathogen in a machine, not inside the body, right? So PCR comes under in vitro gene amplification technique. PCR comes under in vitro gene amplification technique. What do you mean by in vitro? Outside the body. So, if the pathogen is amplified inside the body, it causes damage to the body, right? So, that is why we will multiply the concentration of the pathogen outside the body. So, after that, we go for the diagnosis. So, that is about PCR. So, we are telling it helps in amplification of the gene. It helps in amplification of the pathogen concentration. Now, this PCR stands for polymerase chain reaction, which is developed by Kary Mullis. The 3 steps of PCR are, first one is denaturation. The first step is called denaturation, and the second step is called annealing, and the third step is called extension. The third step is called extension, and the PCR is conducted in a machine called thermal cycler. This is conducted in a machine. What is the name of the machine? The name of the machine is called thermocycler. The name of the machine is called thermocycler. Understood? So I told you in chapter 11 we discussed a complete lecture on PCR. There you can see, so what is denaturation, what is annealing, what is extension and all. Now here we will see like in which techniques we use PCR. One, for HIV detection. Applications of PCR. Applications of PCR. So we can tell that HIV detection can be done by using this technique. So first application, HIV detection can be done with PCR. Nowadays, if you're seeing like RT-PCR they're doing for confirming corona. So means the PCR is also used for detection of dangerous, dreadful diseases like corona also. So for corona detection also, right? So what is that technique called? It is called RT-PCR. It is called RT-PCR. So here coronavirus is a virus in which RNA is there. So reverse transcriptase PCR. This is another thing. HIV can be used to detect genetic disorders also. PCR can be used to detect genetic disorders also. PCR can be used to detect genetic disorders also. It can be used to detect the mutagenic genes which are present in cancerous patients. PCR can be used to detect cancerous genes. Okay, it can be used to detect cancerous genes or the mutated genes in cancerous patients. Cancerous genes, or we can write it as mutated genes present in cancerous patients. Mutated genes present in cancer patients. So there are wide applications of PCR. We have listed only those which are mentioned in NCERT. So NCERT says that PCR comes under molecular diagnosis. So PCR will amplify the sample outside. In vitro amplification it will do, not causing any harm to the person. PCR helps in early detection. So HIV in early detection helps to save the person, right? It helps to treat the person in a better manner. So it is helping in HIV detection, it is helping in detection of corona, it is helping in detection of genetic disorders, and it is helping to detect mutated genes which are present in the cancerous patients. All this is about PCR. Now let's talk about another technique. What is that? Autoradiography. Take a screenshot. Right, now let's talk about the second technique which comes under molecular diagnosis, and that is called autoradiography. Now, just now we are telling that, so it should be a normal gene, and when it gets mutated, it becomes cancerous. Now we want to identify whether the gene is normal or it got mutated and it became a cancerous gene. For that, we can use a technique called autoradiography. So the second tool or technique under molecular diagnosis is autoradiography. The technique is called autoradiography. So what will we do in this autoradiography? We want to find whether it is a normal gene. By using this technique, we can find out whether it is a normal gene or it is a mutated gene. If it is a normal gene, then the person is healthy. If it is a mutated gene, then the person is having the disease, right? So we want to find out whether they are having normal gene or mutated gene. For that, what we need to do? So we know after human genome sequencing, we know the sequence of all the genes, right? So what is that ATGC sequence? That we know. So to that normal gene, can we prepare a probe? Can we prepare a complementary radiolabel probe? Yes, you can. If the DNA is having a sequence of AAA, so you can prepare a probe with TTT, and in that probe means it is a radiolabeled particle. In that, wherever phosphate is there, ATP, GTP, CTP, TTP will have phosphates. Wherever phosphate is there, radiolabel with P32. So that means we can prepare single-stranded DNA or RNA tagged radiolabel probes. So we will go with single-stranded DNA or single-stranded RNA tagged with radio labeled probes. For example, we want to identify a gene, a normal gene, in which the sequence is G, 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 right? Now, if it gets mutated, let's see that also. If the gene gets mutated, then it got converted to C, C, G, G, G. So, two bases I changed. The first two C's I changed into G's. Okay, or else we can write C also, we can write T also. For better clarity, we'll take T. C got mutated, G got mutated to T. Now, we want to identify whether it is a normal gene or a mutated gene or defective gene. For that, we need to prepare a single-stranded DNA as a probe particle. So, we will take a single-stranded DNA. We know this sequence. After human genome sequencing, we know this sequence. So, I am preparing a probe of C, 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 C. And this probe, it is CTP, 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 CTP. Now, CTP, phosphate it has. So, use radio label phosphate here. So, here, I tagged this. What is this? P32 radio label single-stranded DNA or single-stranded RNA, a small single-stranded DNA or RNA which helps in identification of the gene is called as a probe. Probing means checking, right? So, this one is called as a probe. The probe which is having radioactivity. So, we can call it as a radio labeled probe, right? It is called as a radio labeled probe. Now, add this radio labeled probe to the gene. Now, if the person is healthy, then the gene is as it is, like G, 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 G. Then when you give the radio labeled probe, then yeah, the radio labeled probe can go and bind, because it is a complementary sequence, C, 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 C. It will bind. If it binds, then how can you check whether the double-stranded DNA is made or not? So, we can check with a technique called autoradiography. What is autoradiography? Means we will trap, it gives radiations. Radio label particle will give radiations. You can trap the radiations on an X-ray photographic film. So now we will take an X-ray photographic film. I think all of you might have seen X-rays, right? So the way the X-rays pass and they give the images, we have seen that. In the same manner, if there is a radio label particle here, this radio label particle will give an image on an photographic film. X-ray film we call it as X-ray film or photographic film. On the photographic film, it gives the radiations like this. The radiations will be trapped on the photographic film or X-ray film if the probe is going and binding. If the probe is going and binding, if at all the gene got mutated, if it is a cancer and the gene gets mutated, so T, T, G, G, G is there. Now your probe is CCCs, right? Can it bind here? It can't. It can't. So means the initial sequence itself is different, so it cannot bind. So here, no probe attachment. When the probe is not attaching, then no image on X-ray film. There's no image trapped on the X-ray film. So such a simple technique, see, we can use it for identification of the presence of a pathogen and all. What is this technique called? Autoradiography. In autoradiography, what are we taking? We are taking a radio label probe, which is having complementary sequence against a normal gene. If the gene gets mutated, then it cannot bind to the radio label probe. When it cannot bind, you cannot trap the image on the X-ray film. That's it. Take a screenshot. Then we'll talk about the third molecular diagnostic technique called ELISA.